Live?
It's
Yeah.
live, Aditya.
Yeah. Go, Aditya.
Okay. So for the next talk, we have Dr. Ido with us. Dr. Ido Davidesco did his PhD in cognitive neuroscience from the Hebrew University of Jerusalem, Israel, and is an assistant professor of learning sciences at Neag School of Education, University of Connecticut, US. His research merges cognitive neuroscience, psychology, and education. He studies how students learn science in real classroom using portable EEG and eye tracking technologies. Currently, he is developing and evaluating science curricula, educational software, and professional development for teachers to enhance classroom-based research experiences for K-12 and university students. Today, he is here to talk about findings from his study on brain-to-brain synchrony between students and teachers to predict learning outcomes. Over to you, sir.
Thank you so much. I'm excited to be here and meet you all. I hope that you're all doing well. I would love that to be as interactive as possible over Zoom, so I'll keep an eye on the chat, and I'm happy to take questions throughout the talk. I also have a few questions to you that I'm going to ask you throughout this talk, so stay tuned. So this talk is about my research with my colleagues at New York University and elsewhere on brain-to-brain synchrony in classrooms. So what motivated this research is that we know very little about the neuroscience of social interactions in general. In fact, this field of research is known as the dark matter of human neuroscience. And the reason is that most neuroscience research is done in highly controlled and artificial laboratory environment, like inside an MRI brain scanner, where people cannot really interact with anyone. They can view pictures or listen to sounds, but it's a highly constrained artificial environment. So in my research, I'm interested in taking neuroscience outside of the laboratory box into the real world, where we interact with each other, typically face to face, but now during COVID, we do that through screens. But nonetheless, we interact with one another, we have conversations that go back and forth, and I'm interested in studying the brain dynamics of people as they interact with one another, and specifically that of teachers and students in classrooms. I find it helpful to think about social interactions in classrooms in terms of joint attention. So joint attention is our capacity to coordinate attention with a social partner. It's clearly very important throughout development. Babies from a very early age can follow the direction of gaze of their caregiver. It's the basis of language acquisition and the acquisition of social skills, but it also plays an important role in classrooms. How many of you have heard the sentence, "all eyes on me," from their teacher? Or perhaps if you are a teacher, how many times have you said that sentence to your students? So when a teacher says all eyes on me, they're basically directing their students' attention to the same target, in this case, their own face. So joint attention plays an important role in classroom learning as well. I also find it useful to think about student-teacher interactions in terms of joint actions. And there is a growing body of literature suggesting that during joint actions, people become implicitly synchronized at different levels. For example, two people on rocking chairs tend to synchronize their movement as if the two chairs are mechanically coupled. Or just like the piano behind me, in a piano duet, a pianist can achieve higher degree of synchronization than the two hands of a single musician, which is quite remarkable. So we have this ability to synchronize our behavior with others in order to cooperate with one another, in order to communicate more effectively. And in my research, I'm asking if this phenomena also happens at the neural level in our brain activity, and does that also happen in classrooms? So our social interactions in classrooms reflected in synchronized brain activity patterns between students and teachers. So that will be the main question that I'll address today. And if so, if we indeed observe brain-to-brain synchrony in classroom, what kind of information can we extract from that? Does that tell us how engaged students are, how effectively they learn, how they feel towards one another, and so on? So this is the outline of my talk. I'll briefly give you an overview about 3 studies that my colleagues and I conducted over the last few years on brain synchrony in classrooms. In the first study, we focused primarily on student engagement. In the second one, on learning outcomes. That is, can we predict how much information students retain from a lesson based on their brain synchrony? And the third one, which is still ongoing, is about collaborative learning. It's about students learning in small groups. And if I have time, I would also love to share my other line of work, which is about how we can use the same technology to engage students in science learning. And I'll conclude with some future directions. So in order to conduct this research, we cannot use a bulky, expensive MRI brain scanner. Instead, we use portable EEG technology. Now, I want to make it clear that EEG has been around for a long time. In fact, it's been around for nearly 90 years for both clinical purposes like diagnosing epilepsy at hospitals and basic research. What is new or relatively new is our ability to take this technology outside of the lab into real-world environments. And this is due to these more recent developments in portable and wearable technology. So here on the screen, you see some of the EEG devices that my colleagues and I use in our classroom-based research. This device over here is by a company called Emotive. It's a low-cost device. It costs a few $100 US. It only has 14 sensors compared to perhaps hundreds of sensors that you might find in laboratory-based EEG. But unlike laboratory-based EEG, this is a dry system, meaning that you don't need to apply gel, there are no wires, it only takes a few minutes to set it up. And as you can see here, there are also specialized caps for young kids. I have some new studies where we look at parent-infant synchrony. So for that, we need to acquire EEG data from young infants. These are the most advanced devices that I'm currently using in my research. They have more channels, 32 channels. But one important thing to note is that a major limitation of EEG is that it's highly sensitive to movement. And that's a major concern in classroom-based research. So whenever students move their head and talk, that will create artifacts in the EEG measurement. And that's clearly a concern. We take several steps to minimize movement as much as we can during data collection. But of course, we still want students to be able to interact with one another as naturally as possible. And then also when we analyze the data, we take many steps to ensure data quality. But all in all, the quality of the data coming out of these portable devices is definitely lower. It's not as good as what we can obtain in a laboratory setting. Okay, so here's a quick video from the very first study led by my colleague Susanna Dicker that we conducted in New York City a few years ago. So this is a small group of high school students wearing EEG devices. As you can see here, students were helping each other, putting up the devices. They were able to observe their brain activity on the screen, which was a very engaging experience for them. And then when everyone were set up, the teacher and the students, we started collecting data. At first, just some baseline data when the students were just sitting quietly. And then the teacher started teaching, and we looked at different classroom activities. So all in all, we had a small group of just 12 students and their teacher, but we measured their brain activity across 11 classes distributed over a semester. So we collected a good amount of data from these students, excuse me, and as mentioned, we looked at different instruction activities where the teacher was just reading aloud from his notes or presenting a short instructional video, giving a lecture, or facilitating a group discussion. And these activities were repeated throughout the semester multiple times. So the first question that we wanted to ask is, can we say anything about how engaged students are in these activities based on their brain-to-brain synchrony? So just very briefly, I'm not going to get too technical, but I'm happy to answer more questions about the methods at the end. How do we compute brain synchrony? What does that mean? So in simple terms, you can think about brain synchrony as the correlation between brainwaves. So to what extent the brain activity of different individuals go up and down in sync. In order to do that, we first decompose the EEG signal into different frequency bands. So you may have heard about brain activity in the delta or theta or alpha range. So these are discrete frequency bands that people usually use in EEG research. So we can decompose the EEG signal into slow oscillations in the delta band all the way up to a much faster activity in the beta band. And then within each frequency band, and primarily within the alpha band, because there is prior research suggesting that alpha activity is related to attention. So within each one of these frequency bands, we computed the synchrony between one student and each one of the other students in the group. So this is a pairwise measure. It's computed for a pair of students, and then we can average all these values into one measure that we call student to group brain synchrony. And that will be the main dependent measure throughout this talk. That is, to what extent this specific student, to what extent their brain activity is synchronized with the brain activity of their peers. So in order to look at student engagement, we used a survey that was administered at the end of each class session, where we ask students to rate their engagement level on a scale. And the results here might not be very surprising to you. So students were more engaged by instructional videos and group discussions compared to lectures and readings. What was quite striking was that we observed the very same pattern of results for student to group brain synchrony, where brain synchrony was significantly higher during videos and discussions compared to the other two activities. And even more importantly, these two measures, student to group brain synchrony here on the x-axis and self-reported engagement here on the y-axis, were significantly correlated. So brain-to-brain synchrony predicted self-reported student engagement. The more engaged students were, the higher their brain synchrony with their peers. And here's how we think about these results. So when students are not engaged in a task, when they're distracted, when they mind wander, we know based on prior research that there is an increase in their alpha brain activity. I already mentioned that alpha brain activity is associated to attention, but this is an inverse relationship. So when engagement goes down, alpha activity goes up. So you see how we have these high amplitude alpha oscillations, and these alpha oscillations are idiosyncratic and they're not synchronized across students because each student in this case is, let's say, mind wandering and think about completely different things. However, when students are highly engaged in a task, as I hope you all are now during this talk, alpha oscillations are suppressed. And that allows a much higher degree of synchronization between students. So under high engagement, we expect to see high brain-to-brain synchrony. I'm going to stop for a sec, and I'm happy to take any questions, as I mentioned, throughout this talk. So feel free to either type a question into the chat or just unmute yourself and ask in your own voice. So are there any questions so far?
I have a question. We have emotives, but when we used it a while ago, because there are only 14 channels, the data was not kind of a research trends in that sense. I mean, we were able to see the differences between the electrodes itself and for things like opening eyes and closing. But when listening to music or something, we were not able to look at the differences at any of the frequencies bands that you're talking about. How were you able to do it?
Yeah, so the results that I just shared with you were actually obtained with the emotive devices. Oops. So I agree, the data quality is not great, but the data in our experience is usable. However, in the study that I'm about to tell you about, we actually use more advanced technology. So this device that you see here is by a company called Neuroelectrics. And this cap has not 14, but 32 EEG channels. The sampling rate is higher. The data to noise ratio is better. You still get movement artifacts, but perhaps slightly less. So yeah, I agree that using emotive can be challenging. We primarily use emotive now for teaching purposes. So that's this other line of work that I hope I'll have time to get to. But thank you for asking that. That's a good point.
Okay. There's one more question in the chat.
Yeah, so that's an interesting question. Do higher amplitude and variability of alpha go together? So if you're referring to variability between individuals, the answer is yes. So let me go back to my other slide. I think that this question is based on this slide. Yeah, so when students are not engaged, we see an increase in alpha brain oscillations. So we see higher amplitude alpha waves. And these waves tend to be variable across individuals. And that's why under low engagement, we don't see high brain synchrony. So I would say that, yeah, high amplitude in this case goes hand in hand with higher variability across students. Can I say higher brain active area, higher the frequency? Well, not necessarily. So, oh, I'm sorry. Yeah, do you want to clarify or ask another question?
No, go ahead. I think somebody just unmuted their mic, so just go ahead.
Okay, yeah. So you'll find these different oscillations pretty much everywhere in the brain. It's true that different brain regions might have different dominant frequency, at least at rest. But for instance, during sleep, the entire brain pretty much is switching from alpha beta, which is what we see in wakefulness, to slow wave sleep, which is delta. And during slow wave sleep, where delta is dominant, that doesn't mean that the brain is not active. So neurons are still very active during slow wave sleep, but the difference, and this is interesting, what creates these high amplitude slow waves is that many neurons are synchronized. So they all fire at the same time and they all become silent at the same time. And that allows their electrical signal to sum up to create these large amplitude waves that we can measure from the scalp. So slow brain activity doesn't necessarily mean low brain activity. I hope that that makes sense. Any other questions? These are great questions, so keep them coming. Okay, so yeah, if there are any other questions, feel free to put them in the chat at any time. Oh, here's another one. Would you consider monitoring the brain activity during a self-study or video tutorial? Oh, that's a great question. In fact, I just submitted a grant proposal with some colleagues where we plan to look at how students learn with a cognitive tutor, with what is called an intelligent tutoring system. So that will be just an individual student working with a computer where they go through a tutorial, they are asked questions, and we plan to measure their brain activity with EEG and also their eye movements to supplement data that is coming out from the tutor. So the tutor is collecting data, behavioral data about the students, how many questions they answer, how much time it takes them to answer these questions, are their answers accurate or not, and we plan to basically triangulate this data with brain data, with eye tracking data. So I don't have any results to share with you, but perhaps next time we meet, I'll be able to talk about that study. So thanks for asking. Okay, so far I talked about engagement, but we also have an interesting set of results about social dynamics. So if you recall from the video that I shared with you earlier, we had this condition where the students were sitting quietly, making eye contact with one another. So we asked these students how close they feel to one another. We asked that at the very end of the study. So perhaps a couple of months after the EEG data was acquired. But nonetheless, we saw a significant correlation between the degree of student-to-student brain synchrony and their self-reported closeness. So pairs of students who reported higher mutual closeness, now, mind you, these closeness ratings might not be reciprocal. So perhaps a student A might feel very close to student B, but that student B might not feel as close to student A. So we looked at the average closeness. And the average closeness, the higher the average closeness between two students, the higher their brain-to-brain synchrony during this eye contact period that we had before each class session. Now, mind you, this is a small sample of students. Clearly, these findings need to be replicated, but there are also recent findings using functional MRI that also point in this direction. So this reference over here by the Whitley group at Dartmouth College, that's an fMRI study showing that students who are closer to one another in their social network show more similar brain activity patterns than students that are further away. So close friends show more similar brain activity patterns than complete strangers. So brain-to-brain synchrony predicted not only engagement, but also social closeness. And in a follow-up study that we published in 2019, we actually extended that to student-teacher closeness. So we, in that other study that was done in another school, different teacher, different students, we looked not only at student-to-student brain synchrony, but also at student-to-teacher brain synchrony, and demonstrated that students who felt closer to the teacher showed higher brain synchrony with the teacher. But what about learning? So far I talked about engagement and social closeness, but do students learn better when their brains are in sync? So that was the main question that we addressed in the second study, together with my very talented research assistants, Emma Laurent and Henry Valk. This was not a classroom-based study, that was a lab-based study where we tried to simulate a small classroom. We invited 4 students at a time, and they interacted with a teacher. And these were undergraduate students, so college students rather than high school students. The reason is that at that point at that point we just acquired these more advanced EEG devices, the neuroelectrics devices that I mentioned earlier, and we wanted to test them out in the laboratory before we go out to schools. And other studies, again, there are very few studies that looked at brain synchrony, but one of our previous studies showed that surprisingly, there was no association between synchrony and learning. And there was another study from another research group that actually showed that when students watch instructional videos, there is a correlation between synchrony and learning outcomes. So that really motivated this study to try and figure out what's going on. So we had students coming to the lab actually a week before the EEG session for a pre-test. We really wanted to measure their learning very systematically. So we asked them a set of questions about the material that they were about to learn a week before the EEG session. And then during the EEG session, they went through a sequence of 4 mini lectures on different topics in biology and chemistry. You can see the specific topics over here. And immediately after each lecture, they took a post-test where they were asked the same set of questions about the topic that they just learned. And since in education, we want students to be able to retain information over time, we also invited them back a week later for a delayed post-test where we didn't measure any EEG, we just gave them the same test to look at slightly more longer-term learning. And here we also had a much larger sample of students. We had 31 students. As mentioned, they came in groups of 4. And if you do the math in your head, the math doesn't work because we couldn't include all the data. Some students didn't provide a good enough data and had to be excluded, but this was a small minority. And the first set of results that I'll show you is just how much students learned. So these are the test scores across these 3 time points: a week before, immediately after each mini lecture, and a week later. And you see that a week before, these are, I should have mentioned, these are non-science major students, primarily psychology students. So their knowledge about chemistry and biology was quite low. Clearly they had some background from high school, but these are not students that have much background in these topics, which is good because that allowed this big delta between the pre-test and the immediate post-test. So there was a big improvement in their test scores. And as you can see, a week after, there was some decline, which is to be expected. And the main outcome measure that we were interested at here was this difference between the delayed post-test and the pre-test. And this is what we call learning gains. Yeah, so good question. The order of the topics was indeed randomized. Yes, so each time we started with a different topic to control for any order effects. Thanks for asking. Okay, so I mentioned that I'm going to have some questions for you guys. So the question that we were hoping to address here was not only whether we can predict learning outcomes based on brain synchrony, but how the predictiveness of brain synchrony compares to self-reported engagement. So just like in the first study, we asked students immediately after each lecture how engaged they were on a scale from 1 to 7, 1 being low engagement, 7 being high engagement. And we were interested in to what extent we can predict learning gains based on these self-reported engagement measures. We also looked at intrabrain synchrony. So just like computing synchrony across individuals, you can also look at synchrony within individual brains between different EEG electrodes. So to what extent, let's say, the front of my brain is in synchrony with the back of my brain. That would be intrabrain synchrony. And finally, we also looked at brain-to-brain synchrony. And as you can see, we primarily looked at alpha because of this prior research on alpha and attention, but we also compared that to other frequency bands. So here's my question for you, and feel free to use the chat if you'd like, or just think about it for a moment. So what pattern of results would you predict? What do you think is a significant predictor of learning? Self-reported engagement, intrabrain synchrony, and or brain-to-brain synchrony. So I'm going to take a sip of water and let you think about it for a sec. Okay, so I see some hypothesis in the chat that it might be brain-to-brain or intrabrain. Of course, it can be both or all three. It doesn't have to be just one, yeah? So let's see. So surprisingly, surprisingly, there was only a trend, but it wasn't significant between self-reported engagement and learning gains. So we predicted based on prior research that the more engaged students are, the better they learn. But here there was only a trend, it was not significant. And we can talk more about why that might be the case. With intrabrain synchrony, so when we measured synchrony within individual brains, there was no correlation whatsoever with learning. So the two were not associated with one another at all. However, brain-to-brain synchrony was a highly significant predictor of learning, where higher synchrony, specifically in the alpha band, not in other bands that we looked at, significantly predicted learning gains. So students who showed higher synchrony with one another, and also with the teacher, that's not shown here, but we also looked at the teacher, showed better learning a week after the EEG session. And we also included all these measures in one big statistical model, showing that brain-to-brain synchrony predicted learning above and beyond what can be predicted based on just engagement or intrabrain synchrony. But we also wanted to take a step further and ask if brain synchrony can not just predict learning outcomes overall, can it also predict how students answered individual questions in the test? So in order to do that, we took the lectures and we transcribed them. Mind you that these were live lectures, they were not recorded. So each time the teacher delivered the lecture, it was similar because they used the same slides, but not identical. So we had to transcribe all these lectures and identify when in the lecture each question was answered, or at least when the critical information was provided. So here's an example. So this is one of the test questions on bipedalism, how come we stand on 2 feet? So which of the following is evidence that bipedalism was not exclusively an adaptation to the drier savanna ecosystem? And the answer to this question was provided within this time frame. in this specific case. So rather than computing brain synchrony across the entire duration of the lecture, now we computed brain synchrony only within this time window and asked whether that can predict whether the student answered that specific test question correctly. And here are the results. So overall, alpha band brain-to-brain synchrony was significantly higher for test questions where students showed learning, that is, questions that they answered incorrectly before the lecture and correctly after the lecture, compared to questions where no learning was observed, to questions where their answers did not change. And I believe that this is an important finding because it suggests that brain synchrony can provide information about specific moments in a lesson and about whether students can retain this information over time or not. So once again, I want to stop, and I'm happy to answer any questions about the second study or anything else that I covered so far.
So are these questions, I mean, the answers written, I mean, did they type in the answer or was it multiple choice? How did you know what is correct? I mean,
yes,
exactly.
Great question. So these were primarily multiple choice questions where there was only one correct answer. We also had 2 open-ended questions at the end, but they were not included in this analysis. And of course, you can argue how accurately we can capture students' understanding with these multiple choice questions. That's a legitimate concern. So in terms of the type of students that we worked with, so these were undergraduate students at New York University. I wouldn't say that they were an elite group of students, but it was a relatively homogeneous group, right? So they were all primarily psychology students, undergrads at a single institution. So clearly we didn't have much diversity here. Although if you look at the test scores, you do see some variance, right? So these students, for instance, came into the study with a much better prior knowledge than that student. So they were not all at the same level in terms of their prior knowledge about these science topics. Any other questions?
In this distribution that you have of people who got it accurate, the test scores, there are a lot more outside your standard deviation than in, right? Especially for the pre-test and for the delayed post-test. How do you account for that huge variation?
Yeah, that's a really important observation. Yeah, so indeed the variance in the delayed post-test was much greater. And this is interesting, right? This basically tells you that even though they pretty much started at this, well, not the same level, but there was less variance in their knowledge pre-test, in the post-test there was greater variance. So some students benefited more from this lecture, so retained more knowledge than others. And we accounted for that by looking at this delta at the individual student level. So for each student, we computed the difference between their post-test and pre-test. And clearly for some students, this difference was large. Let's assume that this is an individual student where they scored at 20% pre-test and at 80% post-test. So this is clearly a large gain, 60%. For other students, the gain was much, much smaller. Some students even went down. So their knowledge was actually slightly higher during the pre-test. Perhaps they just guessed the right answers in the pre-test. And we definitely want to look at that more closely in future studies. Yeah, so the mechanism behind brain-to-brain synchrony, that's a great question. I wish I had a good answer to that. I'll get to that in just a few minutes. And is it also possible that there were differences in rehearsal? Yeah, for sure. So we ask students not to look up the material online. But perhaps some students did that nonetheless. It wasn't a classroom-based study where students learned for this test. We didn't expect them to learn. They didn't have any written materials to learn from. So they came into the lab, they received all this information, they went home, and then a week later we tested them again. So it wasn't, again, it wasn't an organic group of students. So I don't think that rehearsal had a big role in what we see, but I cannot, of course, completely rule that out. Okay, so let me briefly tell you about the third study, which is still ongoing, and then I'll get to the mechanism question, and perhaps we'll pause there again. So the third study, which is still ongoing, is about collaborative learning. There is a huge body of research suggesting that overall collaborative learning is more effective than individual learning, but we don't really know how and why collaborative learning works. And as teachers, I'm sure that you had the experience of sometimes collaborative learning doesn't work very well. Sometimes groups do not work effectively together. And the question is why? So to address that, we conducted another classroom-based EEG study with high school students again. But in this case, we had students work on a task in a small group. So their task in this case was to build a model of a cell. We gave them a mini lesson, just a refresher on cell biology, and then we gave them some arts and crafts materials and asked them to build a model of a cell as a group. They were given about 10-15 minutes to complete this task. So let me show you a quick video of just one group of students. And my question here for you is, what did you observe, or what you are about to observe, about how the students interact with one another?
Oh my God, we fully have to draw
This
out.
is the endoplasmic reticulum, these are the ribosomes. This is the nuclear membrane, this is the nucleus.
Bio is getting next year. Oh my god.
Wait.
Yeah, I know,
So
I'm doing that for a reason.
should we use? So what do we have? We have the button as the cell membrane. Okay.
So we need to see this all around.
So I guess just the button. Well, I mean, they can understand that the buttons were.
For the nucleus.
Okay, I hope that you could hear the sound. So you may have observed that these two female students in this case seemed to be much more engaged and active than the two male students. Specifically, this student AH, she was the recorder, she took notes. So she kind of took the leadership role here. And the question that we're now looking into is how these highly complex dynamics within this small group of students, how is that reflected in their brain activity? So one thing that we've already done was to take all the videos, so we have video recordings of all these sessions, and code them. So we asked for each individual student what they were doing at any moment in time. Were they just idle, meaning seemingly not engaged? Were they physically engaged with the materials? Were they just verbally engaged by talking about the task? Were they both verbally and physically engaged with the task? Or did they seem to be off task? So as you can imagine, these are high school students. So at times they were just completely distracted talking about their weekend plans. And what this graph shows you is the percentage of time where students basically manifested these different types of behaviors. And you see that overall, for the most part, students stayed on task. But there was a good a chunk of time, about almost 15% of the time where they were off task, and about just over 15% where they seem to be non-engaged. But of course, when students seem to be non-engaged, so when they're not doing much, we don't really know what is going inside their heads unless we use EEG. So this is just some preliminary data showing that the percentage of time when students were idle was associated with EEG power in the alpha band, the same alpha band that I was talking about earlier. So there seems to be some interesting data to explore. We're still looking into this data. But the part about this study that I'm mostly excited about is looking at EEG activity and specifically at brain-to-brain synchrony in correspondence to student conversations. So here's a segment of a conversation that the same group of students that we just looked at had just after this video that I presented earlier. So in this conversation, IB, this male student, actually starts speaking. So he was not silent throughout the entire session. He held a pipe cleaner and said, "There, I made another endoplasmic reticulum." And a second later, JA, the other male student, says, he picks up the figure and says, "Should we put the ribosomes on it?" And AH, again, she was the recorder, she agrees, she starts writing. But then at this point in time, she puts down her pen and announces, "I'm so tired, I really need to leave." And this really derails the conversation because from that point onward, they started talking about how many hours of sleep they got the previous night. Not bad for IB for getting 10 hours of sleep. So this moment in time is interesting because that's a switching point from on task to off task. And the question here is, how is that reflected in students' brain activity? So one thing that we're trying to do now is to look at how brain synchrony unfolds over time and identify moments of high brain synchrony and low brain synchrony based on the EEG data, and then go back to the video. And one prediction is that high brain synchrony would be characterized by high joint engagement where all students are focused on the same task, whereas this is how this group of students might look like during low engagement. But I think that what makes this research exciting is that we don't really know what we're going to find. So again, we don't yet fully understand brain synchrony, and we might learn new things about what kind of behaviors lead to high synchrony and in turn to better learning in group settings. So what I want to conclude with, and then I'm happy to open it up again for discussion, is what is brain-to-brain synchrony? What is the mechanism? And the short answer is that we don't really know. It's a new line of research. We don't yet have a good understanding. We know that at the most basic level, brain synchrony is generated simply by the fact that the students are exposed to the same input. So they're all watching a video together, they're all listening to a lecture or engage in a group activity together. And that by itself can generate brain synchrony just because we're all presented with the same material. But my data and my colleagues' data suggest that brain synchrony is modulated by attention. So when students are more engaged in a task, they show higher brain-to-brain synchrony. And it's also modulated by learning, or at least predicts learning. So when students show higher brain-to-brain synchrony, they also tend to do better on a post-test. So brain synchrony seems to be a highly complex, multidimensional phenomena, which is partially driven just by bottom-up stimulation, but it's also modulated by attention, by social closeness, and other factors. And perhaps I'll just briefly mention that this phenomena is not unique to humans. In fact, there are some recent studies documenting brain-to-brain synchrony in animals, in this case in bats. So these were 2 bats flying around, socially interacting, clearly in a very different way than what I just showed you in a classroom. But nonetheless, these bats showed correlated neural activity patterns at different time scales, from seconds to minutes to hours. And the degree of brain synchrony between these bats corresponded to their social interaction. So when they were truly interacting with one another, they showed higher brain-to-brain synchrony. So I'm hoping that you are now at least intrigued by this phenomena as much as I am. And clearly we need more research to really pinpoint what generates brain-to-brain synchrony and understand better how we can use this information in a classroom environment. So since I want to make sure that I leave enough time for questions, I'm going to skip the part where I was about to tell you about how we can use this EEG technology to teach neuroscience in classrooms. If you're interested in that, I'm happy to tell you more about it and also drop a link to our website. But I do want to acknowledge the many different people who were involved in this research, primarily David Poppel and Susanna Dicker from NYU, the Department of Psychology, and many other colleagues who contributed, as well as my funding sources. Thank you for your attention. And once again, I'm happy to take any questions.
Thank you, Dr. Ido. I'll just wait for questions before I ask. Questions? So before, I think somebody might type. So I just wanted to know, do you do face, face corrections when you're doing this synchronization? Frequency phase analysis?
Yeah, that's a really good question. So there are different ways to look at brain-to-brain synchrony. And some of these methods are, can be biased. So there is some research showing that some methods might lead to spurious synchrony, where it seems like there is synchrony, but in fact there is not, just due to the way that we compute it. So we are very mindful of that. In the most recent studies, we use a measure called circular correlation. And this is a phase-based measure. So indeed, it looks at the phase consistency across individuals. So rather than looking at the amplitude of their brain activity, we look at specific frequency bands, and then we compute the instantaneous phase, and we ask to what extent the phase is consistent across individuals. And that seems to be a more robust measure of synchrony, which is less, well, we cannot rule it out, but it's less likely to be influenced by spurious synchrony across individuals. Yeah, I see a question about science education, so thank you for asking that. So I just dropped a link in the chat to our Brainwaves curriculum. So unfortunately, I don't have time to tell you about it in depth, but the website has much more information. Briefly, this is a high school neuroscience curriculum where we use the very same portable EEG technology, these emotive devices that I showed you earlier, but this time not to collect research data from students, but instead to teach them about the brain and let them become the researchers. So students work in small groups, mentored by a scientist on developing an EEG study. And they come up with some really interesting experiments related to how the brain processes visual information and auditory information. Topics like music and video games are, as you can imagine, are very popular in this population. And the goal is not just to teach students about the brain, which is an important goal by itself, but also to expose them to scientific research more broadly. So it's called Brainwaves, and there is more information on the site, including our full curriculum, which is publicly available.
I have, if anybody else wants to ask a question. I have one question. Yeah, you talked about the engagement of the students, right? There could be a possibility that somebody is not physically talking, not acting, not moving, but they are still in the task. How do you kind of take that into consideration?
That's a fantastic question. I think that that's really the power of this method, because the way that we usually, let's think about collaborative learning as an example. So the way that we typically study collaborative learning in classrooms is either by observing students or by asking them questions. And these are important, useful methods, but they're limited, right? Because students don't necessarily know how to accurately report their engagement level. Also, you probably don't want to ask students how engaged they are every minute, because that by itself will be very distracting. And with classroom observations, this is less intrusive, right? You just put a video camera and hope that students wouldn't mind, but it's limited to student explicit behaviors. And as you just mentioned, the student might be deeply engaged in a task, but look completely disengaged, and vice versa. And I think that that's really the power of these portable technologies, not just eye tracking, but also, sorry, not just EEG, but also eye tracking, because they really allow us to start looking at what is happening under the hood. And that's what we're trying to do now in this collaborative learning study. We specifically look at time periods where students seem to be idle, so they don't do much. They don't speak, they don't interact with the materials, and yet it seems based on their brain activity that they are deeply engaged with the task. So that's what we're trying to start looking at now. Clearly, it's still preliminary, but I find it a really exciting direction for future research.
There are two more questions, I think, for
Sure.
time for it. So
yeah, so let me answer these questions quickly. So what is the role of the teachers in influencing brain synchrony or brain activity more generally? Great question. As mentioned, we don't know enough about these phenomena to be able to suggest any practical implications. We're not there just yet. I wish that based on this research, we could improve the way that we teach in schools, but as of now, it's just a basic research tool. But I would say that when teachers are able to engage those students effectively, that's when we expect to see high brain synchrony between students and between the student and the teacher. So I think that the main thing that we're measuring is probably a joint engagement, and effective teachers are clearly very talented in getting their students engaged, but of course there are limits to that, right? So with these tools, we can start asking questions about how long can students stay engaged on a task, and what kind of instructional methods can help students stay engaged, and how is that reflected in their brain activity. And very quickly, I'll try to answer the other question. So was there any study done with a variety of teachers and teaching methodology? So storytelling, fun, music, how does that affect engagement and brain synchrony? Yeah, another great question. So as mentioned in the very first study, we looked at 4 teaching activities, nothing as creative as what you listed, but we looked at least at reading, lectures, videos, and discussions. And as mentioned, we observed higher brain synchrony for videos and discussions. But I agree, there are many other teaching methods that we can look at that's still a direction for future research.
yeah, thanks a lot. I think on behalf of all the participants, very interesting talk. It's very new, and also it kind of opens up a lot of venues for us to explore. A lot of information, a lot of interesting experiments can be done. It's still new, but it's a lot of interesting experiments. Thanks a lot for taking the time to give us this wonderful talk. And on behalf of all the participants, again, we thank you. and hope to connect with you sometime soon.
My pleasure. And I just dropped my email in the chat. Feel free to reach out via email for any additional question. Thank you so much for inviting me.
Thank
It's been
you.
a pleasure.
Bye.
Take care.
Shrikant, you can turn it off.
Ma'am, I think you became the host. Can you make me host? Hello?
Yeah, yeah.